Hey guys, and today we're going to learn the first chapter of IB biology, which is known as cell biology. Let's begin. So, before you start off any chapter, it is always recommended that you look at the subject guide that the IB provides. It gives you a great idea about the important concepts that you have to know in the chapter, and the different things that you have to look out in the chapter that will be tested in the exam, or they have the potential of being tested in the exam. So, they give you an idea about the nature of science, which links to your TOK section of your paper, understandings, which is the concepts that you need to know in this topic, and that particular unit itself, international mindedness, which comes as a form of application, which they can often ask in your essay questions, theory of knowledge, utilization, which also comes under international mindedness. You also have application and skills, guidance, and aims. So it's always amazing to have a look at the subject guide, and it serves as a great revision um, look back as well when you are looking to revise anything in that particular chapter. At least you know what you have to look out for, and then that way you can strategically plan your revision as well. So let's begin. Introduction to cells. So you start off learning this chapter by getting acquainted with the cell theory. So the cell theory has three parts and three important conditions, where it states that all living things are composed of cells. Cells is the smallest unit of life, that means it's a building block of life, and cells only arise from other pre-existing cells. When they say that cell is the smallest unit of life, it means that specialized structures within cells, which are known as organelles, carry out different functions. Organelles cannot survive alone, which means organelles depend on each other to allow the cell to function as a whole, and hence they also survive. And when they say cells only arise from other pre existing cells, it means that cells did not appear from nowhere. They had an ancestral form and then they evolved to form the cells that we know today. So, cells multiply through division. All life is evolved from simpler ancestors, like I mentioned, and you usually have two forms of reproduction, which are either mitosis or meiosis. So mitosis results in genetically identical diploid daughter cells where you have 2n number of chromosomes, and meiosis generates haploid gametes, which are your sex cells, which generate n number of chromosomes, so half of it. So, with the cell theory, of course, you will have exceptions as well. And so, there are three important exceptions that you need to know: the stratiated muscle, the aseptate fungal hyphae, as well as the giant algae. So, let's go through them. The first one, stratiated muscle, it challenges the idea that a cell has only one nucleus. So, if you look at the picture on your right hand corner, you can see a single muscle fiber, and there are all the small little dots are the nuclei inside the cell itself. So muscle cells have more than one nucleus per cell. They are called fibers for a reason because they are very long in length, and they are surrounded by a single plasma membrane. That means they are one big cell, but they just have many nuclei, and hence they do not conform with the standard view that a single small cell has a single small nuclei. Moving on to the next exception, which is aseptate fungal hyphae. So that challenges the idea that a cell is a single unit. So fungal hyphae again are very large in size with many nuclei and a continuous cytoplasm. The tubular system of the hyphae forms a very dense network called the mycelium. And like muscle cells, they are also multinucleated. They have cell walls composed of chitin, and cytoplasm is continuous along the hyphae with no end cell wall or membrane. So it just goes on and on and on. Then we have giant algae. So, giant algae is a single celled organism that challenges both the idea that the cell must be simple in structure and small in size. So, it's gigantic in size for a cell because it's about 5 to 100 mm. So, 100 mm is like 10 cm, so it's like huge. And it's complex in form when you expect cells to be the simplest unit of life in its simplest form, but they are complex in form, and it consists of three anatomical parts, which are your bottom rhizoid, which resembles a set of short roots, a long stalk, a top umbrella that branches off that may fuse into a cap, and single nucleus is located in the rhizoid. So it's not multinucleated, but it is big in size. So going on to the functions of life, so unicellular organisms, which are single celled, are the smallest organisms that are capable of independent life. That means they don't have to depend on other organisms for themselves to survive. So all living things carry out seven basic functions that are integral to survival. So you have metabolism, reproduction, sensitivity, homeostasis, excretion, nutrition, and growth. These are seven basic functions that all living things need to do in order to survive. So your subject guide will make it very clear that you need to know at least two examples of single-celled organisms. One is a heterotroph and one is an autotroph. So in chapter 4 you will learn that an autotroph is an organism that can make its own food, and an heterotroph is an organism that depends on the autotroph or feeds off other organisms for its own survival. So paramecium is a heterotroph, that means feeds off other organisms for its survival. And Syndesmus is an autotroph, that means it can make its own food. So you need to know these two examples because sometimes in MCQ they may ask you about paramecium, they may ask you about amoeba, which is another unicellular organism, and they may also ask you about certain parts. For example, where does excretion take place in paramecium? So you must be able to point out the location where excretion takes place. So, like I show over here, so you need to show that, for example, homeostasis takes place in the contractile vacuole, reproduction in the nucleus, growth, reproduction takes place through binary fission. Because this is a unicellular organism, no need for sexual reproduction to take place. So, mitosis or often binary fission, which is asexual. Metabolism, where it takes place, excretion, where it takes place. You need to know how to label this stuff to show how paramecium shows the functions of life, which are the seven functions as mentioned before. Another very important concept in this chapter is surface area to volume ratio. So, cells need to produce chemical energy via metabolism to survive, and this requires exchange of materials within the environment. So, the rate of metabolism of a cell, which is the function of its mass divided by the volume. So, larger cells need more energy to sustain essential functions, and rate of material exchange is shown by function of its surface area. So a large membrane equates to more material movement. So as a cell grows in volume, it as a cell grows, the volume increases much faster than the surface area, and that leads to a decrease in the surface area to volume ratio. If the metabolic rate exceeds the rate of exchange of vital materials and waste due to the lower surface area to volume ratio, the cell will eventually die because it does not have enough material being exchanged. That means it does not receive enough material to grow, and it is not able to remove all the waste products as well. So due to toxicity or any other factors, it may die. And hence, growing cells tend to divide and remain small in order to maintain a high surface area to volume ratio, so that they can maintain a sustainable metabolic rate and sustainable rate of exchange that is suitable for survival. So, examples that we see in our body, the villi in the small intestine, as well as your microvilli, and you also have your alveoli cells, which help to increase surface area to volume ratio in the small intestine and lungs, respectively. So, emergent properties. So, this is another very important concept in the chapter, which basically says that the whole is greater than the sum of its parts, to put it clear. So, emergent properties arise when the interaction of individual components produce new functions. So, multicellular organisms are capable of completing functions that unicellular organisms could not undertake due to collective actions of individual cells combining to create new synergistic effects. So cells may be grouped together. For example, cells may be grouped together to form tissues. Tissues are all grouped together to form organs. Organs are grouped together to form systems, and then organ systems are grouped together to form an organism, as shown on the right. So you can see a muscle then becomes a cardiac muscle, which then becomes a heart, which then becomes an entire vascular system, which is also your circulatory system, and then that becomes human. So it goes from cell to tissue to organ to system to organism. Emergent properties. The whole is greater than the sum of its parts. Differentiation. So, it is a process during development whereby newly formed cells become more specialized and distinct from one another as they mature. So, all cells of an organism share an identical genome. Genome is basically the genetic code that codes for proteins universally, and each cell contains an entire set of genetic instructions for that organism. And activation of different sets of instructions or different genes within the given cell by chemical signals will cause it to differentiate. So, gene packaging, this is where it comes in, where within the nucleus of a eukaryotic cell is one in organisms as compared to your prokaryotic, which are, for example, bacteria. So, a eukaryotic cell, DNA is packaged with proteins from chromatin. These proteins are called histoproteins, which you will learn later in the chapter. And so you have two kinds of gene packaging of chromosomes. You have euchromatin as well as heterochromatin. So euchromatin are the active genes that are usually packaged in an expanded form that is accessible to transcriptional machinery. So euchromatin basically means that this is these are the chromosomes that are most frequently used, and hence they are loosely kept within the chromosomes so that they are easily accessible by the different organelles in the cell, so that they can be transcripted and transcribed. And then you also have heterochromatin, which are the inactive genes. That means they're not used as much, and hence, in order to save space, they are typically packaged in a more condensed form, and hence they are not transcribed as well. Another important part that you need to know in this chapter is application. So this is where stem cells come in. So stem cells are unspecialized cells that can continuously divide and replicate. So this is known as the property of self renewal, and they have the capacity to differentiate into specialized cell types, which is known as potency. So there are basically four types of potency: totipotent, pluripotent, multipotent, and unipotent. So totipotent means that they can form any cell type as well as an extra embryonic tissue. So example, your placental tissues when your zygote is forming in the uterus. Pluripotent means that they can form many different cell types. For example, your embryonic stem cells. Multipotent means that they can differentiate into a number of closely related cell types. That means if you have a blood cell, it can your hemopoietic adult stem cells, because a hemopoietic, it gives you the idea of hemoglobin, which you can relate back to red blood cells. That means they can differentiate into red blood cells, white blood cells, platelet cells, but they won't be able to differentiate into, for example, a brain cell or like a skin cell. They'll just remain within that close proximity of cell types. And you have unipotent, which cannot differentiate, but they are capable of self renewal. For example, your skin cells, they won't differentiate to become brain cells or muscle cells. If they are skin cells, they will remain as skin cells. They'll just divide as skin cells and they will renew your skin. So that's how your skin renews, right? Every few days, there's a layer that sheds off, and to self renew, the cells will divide and replicate, and they will form a new layer of skin. So stem cell therapy, with the use of stem cells, they can be used to replace damaged or diseased cells with healthy functioning ones. So this process requires the use of biochemical solutions that trigger differentiation of stem cells into the desired cell type, and then surgical implantation is required of the cells into the patient's own tissue. Because your immune system knows how to recognize which cell is from the body system itself, and which cell or tissue is coming from outside the body, that means it's someone else's, your immune system would naturally attack it. So in order to ensure that your stem cell therapy does not go for a fail and is successful, you need to be able to suppress the immune system's response towards this foreign body, in that sense. So the suppression of the host immune system, to prevent rejection of cells or tissue rejection for that matter, and careful monitoring of the new cells has to be ensured, because these stem cells, as mentioned in the previous slide, they can continuously divide and replicate, and they can differentiate into different cell types. So you want to ensure that they do not become cancerous in that whole um, thing of being able to differentiate and replace damaged cells. So if you can see on your right, you have the different examples of the uses of therapeutic stem cells. So you can use cultured pluripotent stem cells, and you can take it from the bone marrow, for example, to treat leukemia. You can take skin cells to treat burn victims, because a layer of the skin is damaged. So you can replicate the skin cells so that you can form a new layer of tissue on top, and you can replace those damaged skin cells with new ones, so you'll have a new skin. You can take your islet cells. They come from the islets of Langerhans, which is beta cells in your pancreas, which produce insulin to keep your blood glucose level in order. So you can use it to treat diabetes. You also have another treatment, which is insulin, which will be discussed later in the chapters. But insulin, as everyone knows, if you take it via injection, it's very expensive. So they are looking at alternative treatments for patients who can afford stem cell therapy. Myocardium, as you know, the heart muscle is called the cardiac muscle. It is a myocardium, which means it doesn't need external stimulation for the muscles to contract and relax. It does it itself. This will be discussed later in chapter 6 as we go on. But the heart muscle, known as the myocardium, you can also generate a new layer of muscle for the heart. Your retinal cells, which are in your eyes, to treat blindness, which is your Stargardt's disease, which will be um, discussed later on, and nerve cells, which are used to treat Parkinson's or paralysis. So, Stargardt's muscle dystrophy, as I mentioned in the previous slide, the problem is that it affects one in every 10,000 children. So, that means it's actually quite frequent. And it is a recessive genetic inherited condition. That means it has to be passed down generations. And it's a recessive condition, which means you need two recessive alleles in order for the person to be affected by this condition. And mutation causes an active transport of proteins on photoreceptor cells to malfunction. So your photoreceptor cells in your eyes, which are the one that detect light and enable you to be able to see colors and basically whatever is in front of you, start to degenerate. So you practically lose total central vision. So the treatment is that you use embryonic stem cells, and you treat them and divide them and differentiate them into retinal cells, which are then injected into your retina. The retinal cells then attach to the retina and become functional, and then central vision improves as a result of more functional retinal cells. So you're basically replacing all those that got damaged due to Stargardt's macular dystrophy. And the future. So this treatment is still in at the stage of limited clinical trials, but will most likely be used in the future, hopefully. You also have leukemia, which is your cancer of the blood or bone marrow. So it results in abnormally high levels of poorly functioning white blood cells, where your immune system is just attacking itself. So your hemopoietic stem cells, as mentioned in the previous few slides, which were known as multi potent cells, right? They can be harvested from your bone marrow, or peripheral blood, or your cord blood. And chemotherapy and radiotherapy are used to destroy the diseased white blood cells. And then new white blood cells, which are generated from your hemopoietic stem cells, are replaced. And so your hemopoietic stem cells, or your HSCS, are transplanted back into the bone marrow, because your bone marrow is a place where your platelets, red blood cells, white blood cells are produced by the body. So they will differentiate to form healthy new white blood cells, and that will help you tackle your leukemia, which is a cancer. And so the future, these this currently in use, and many people use this treatment actually, and the use of the patient's own HSCS means that there is a far less risk of immune rejection, because you are taking cells that come from within you to generate new cells outside of the body, although, and then put it back into your body. So because your body will still recognize it as own cells, your immune system won't go and attack it. There's a far lesser chance that it happens. Because this is a therapy, there will be ethical issues that do arise from this problem. A lot of the stem cells are actually derived from one of these three sources, which are either embryos, umbilical cord blood, or placenta, and certain adult tissues like bone marrow. So your comparison of stem cell resources or sources can be done through these four factors, which are ease of extraction, ethics of attraction, growth potential, and tumor risk. So as seen on the right hand side of the screen, embryos, they can be easily obtained from excess embryos generated through IVF programs. Your cord blood can be easily obtained and stored, and your adult tissues like your bone marrow are difficult to obtain because they are buried deep within tissues, so it makes it very difficult to take it out. So your ease of extraction is not as much. Ethics of extraction for embryos, they can only be obtained by the destruction of an embryo. So you're almost um, by application destroying an embryo that could become a baby, could become a fetus, so essentially destroying life, if you go through arguments against stem cell therapy. For cord blood, umbilical cord is removed at birth and discarded whether the stem cells are harvested or not. So it's almost like a waste resource, which is very, very useful. And then your adult stem cells, the adult patient can give permission to whether your cells have to be extracted. So there is that factor of consent. inside it. Growth potential for embryos is almost unlimited, whereas for cord blood and adult tissues, they do have reduced potential as compared to embryonic cells. And for tumor risk, because they can grow unlimitedly almost, they have an extremely high risk of tumor development, which can also lead to cancer, whereas it's much lower for cord blood and adult tissues. So ethical considerations on therapeutic use of stem cells will also depend on the source, and which gives the greatest yield as well. So you have arguments for and against therapeutic cloning, which uses stem cells. So, for example, arguments for therapeutic cloning, it gives you a futuristic treatment to treat many different kinds of problems, genetic conditions, cancer, Stargardt's, leukemia. And it's important to have these kind of treatments because there are millions of people that die from these conditions each year. And so, if you can save people with this kind of treatment, won't it be amazing? And stem cells, as mentioned, because one of the sources are embryos, stem cells can be taken from embryos that have stopped developing and would have died anyways. So essentially, because they would have died anyways, rather than letting them go for a waste resource, you're using them to save another person's life, even though that life from an embryo couldn't be developed. And stem cells can be created without the need for fertilizations or and destruction of natural human embryos. So you can induce pluripotent stem cells to almost become embryos. Now, arguments against therapeutic cloning, it involves the creation and destruction of human embryos. So this links back to TOK, where you can ask at what point do we afford the right to life? Like, when do you realize or have the right to say that no, this embryo would have died, so we just use it for a stem cell? And so you're sort of owning that life, and you have ownership over that and control over what survives and what dies. You More embryos are generally produced than are needed, so that means that excess embryos are killed in the whole process. So it means that these embryos could essentially become children, but they were killed because they were being excessively harvested for stem cell therapy. You also have religious and moral obligations and objections, which people call the playing god argument, which they use against stem cell therapy or therapeutic cloning. So these are certain arguments for and against that you need to know for exam. It often does come out as an essay question, 4 marks, sometimes 6 marks, sometimes 8 marks. Really depends when they can ask you discuss the benefits and disadvantages, for example, or advantages and disadvantages, benefits and limitations of therapeutic cloning or stem cell cloning or stem cell therapy. So you also have artificial stem cell techniques, where stem cells can be artificially generated with distinct benefits and disadvantages. So you have two methods, which are your somatic cell nuclear transfer (SCNT) as well as nuclear programming. So your SCNT involves the creation of embryonic clones by fusing a diploid nucleus with an N-nucleated egg cell, and then because you have more embryos being created in this process, it does raise the ethical concern of the exigency of excess embryos. You also have nuclear reprogramming, which means you induce a change in the gene expression profile of a cell in order to transform it into a different cell type. For example, as mentioned in the previous slide, where you can induce pluripotent stem cells to become almost natural human embryos. And this involves the use of oncogenic retroviruses and transgenes, which increases the risk of cancer because you are using oncogenes. And this will be discussed later in the chapter, where your oncogenes are the one that causes tumor formation. So the advantages of somatic cell nuclear transfer, your SCNT, is that it's indistinguishable from embryo-derived cells, while the disadvantage is that it involves ex vivo creation of embryos, which means that more embryos are created than required. And nuclear reprogramming, the advantage is that stem cells are autologous to adult donors, while the disadvantage would be that it uses oncogenic retroviruses and transgenes, which leads to the very high risk of tumor formation and hence by extension cancer. This is an important application that you will see under the subject guide, where you need to know how to label and use and compare different microscopes, which are namely your light microscope and as well as your electron microscope. So your light microscopes, they use lenses to bend light, as the name suggests, light microscopes, to magnify images by a factor of roughly 100-fold. So they can be used to view living specimens in their natural color, and chemical dyes and fluorescent labeling may be applied in order to resolve specific structures. So resolution is not the same as clarity. Resolution means how distinctly you can see different structures in a cell under a microscope or in anything under a microscope. Electron microscopes, on the other hand, they use electromagnets, not light, to focus electrons, resulting in significantly greater magnifications and resolutions. They can be used to view dead specimens in monochrome, although sometimes false color or dyes are used to make your distinctions between organelles better. And you have two kinds of electron microscopes, which are your transmission electron microscopes, TEM, and scanning electron microscopes, which is your SEM. So TEM, which is your transmission electron microscopes, pass electrons through the specimen to generate a cross section of that specimen, while your SEM, which is your scanning electron microscope, can be used to scatter electrons over a surface to differentiate the depth and map the organelle out in 3D. So that's the differences. So take note, one passes electrons, one scatters electrons, one generates a cross section, while the other generates a surface to differentiate depth, and also a map in 3D. So you need to be able to know the different parts of a light microscope, how to label them, and when you must change or adjust certain knobs, such as your coarse focusing knob or your fine focusing knob, in order to improve magnification or resolution. Your total magnification is found when you can multiply the magnification of the objective lens by that of the eyepiece. Your resolution is defined as the ability of the microscope to show two objects separately, which means you show them distinctly, and it is limited by the wavelength of the light. And you also have the resolving power of microscope, which is 200 nanometers. So the magnification, which is an important formula that you should know for this chapter, is size of image divided by the size of object, or also better known as magnification of image over magnification of object in sometimes in cm, depends on what units the question is asking for. So these are simple calculations that you can do, because you need to know how to figure out the magnification of microscopes. Try these out and see if you get the right answer. And this is just a summary of the visible features that you can see under light microscopes. For example, for bacteria, your amoeba, plant cells, animal cells, what are certain things that you will see under light microscopes. So do take note that under light microscopes, when you can see your nucleus, your mitochondria, your chloroplast, cell wall, cell membrane, sometimes you won't be able to see other specialized and much smaller organelles, like your ribosomes and RER, your SER. So for that you will need an electron microscope to be able to see those parts. And with that, this is the end of the first unit of chapter 1, unit 1.1, which was your introduction to cell theory. Thank you so much, and I hope this resource helps you. I will be back with another video to explain the rest of the chapter. Thank you.